Hello, my name is Henne Holstege and I work at the Alzheimer's Center and at the Department of Clinical Genetics at the Amsterdam University Medical Center in the Netherlands. My research interest lies in Alzheimer's disease and then specifically the genetics of Alzheimer's disease. And of course, we want to move on towards structural variations in Alzheimer's disease, which is what my talk will be about. So first, a little bit about Alzheimer's disease. So if you've seen any talk about Alzheimer's disease, then you've probably already seen this picture before. I'm showing it today because I want to show that, in the process of Alzheimer's disease, holes appear in the brain. So whatever cure you design, that will never bring back the cells that used to be there. So really the only cure for Alzheimer's disease is preventing it. But to prevent Alzheimer's disease, we need to know on forehand, so we need to predict who will get Alzheimer's disease at a later age before symptoms occur. And preferably also which type of Alzheimer's disease or which type of vulnerabilities this person has, so that we can correct that with personalized treatment strategies. One of the tools that we have in our hands to predict who will get Alzheimer's disease is the genome, because based on twin studies, we know that about 60 to 80% of Alzheimer's disease is genetic. And in fact, both ends of the cognitive spectrum is genetic, because escaping Alzheimer's disease until at least 100 years is also genetically defined, at least for 60%, based on sibling relative risk. So we can really use the genome to predict who will get Alzheimer's disease and who will not get Alzheimer's disease to a really great extent. The problem is now that we do not yet know all the genetics or the genetic variations that are associated with Alzheimer's disease. So therefore, I will show you what we do know by showing you a short history of Alzheimer's disease, and that we have not been able to look yet in the structural variations of Alzheimer's disease, and that we should be doing that in the future. So first, a short history of Alzheimer's genetics. So between 1990 and 2000, we were doing linkage studies. So we were looking for really large pedigrees like this one, where Alzheimer's disease is autosomal dominantly inherited with in a pedigree. So and that's something that you can really see here. This is a presenilin 1 mutation, and the interesting thing here is that when you carry a presenilin 1 mutation, the age at onset of the disease is very, very early. So it's very, very easy to distinguish a person with early onset Alzheimer's disease from the presenilin 1 mutation, to distinguish it from late onset Alzheimer's disease, which of course occurs a lot more often. So there were 3 genes identified using linkage analysis, and those were the amyloid precursor protein, the presenilin 1 gene, and the presenilin 2 gene. However, together, these genes explain about only 10% of the families with autosomal dominant Alzheimer's disease. So that means that there are a lot of things, or a lot of genetic variants that we simply do not yet know, and which we really need to know to understand this disease better and also its etiology better. So to find out more of these genes or genetic variations, the field turned to GWAS studies, so genome-wide association studies, where we use common SNP markers to identify genetic haplotypes that are associated with disease. So this is how that works. Genetic haplotypes are scattered throughout the genome, are printed on an array, and it's very, very cheap to hybridize an array so that you can genotype all of these markers, and that will allow us to identify haplotypes that are associated with the disease, that occur more often in people with Alzheimer's disease than without Alzheimer's disease. And this is what you can then see. So these are actually all of these markers that are spread throughout the genome, and there are then blocks of SNPs that are in linkage with each other that then turn up. We call this a Manhattan plot. So we see all these genes, but actually these are regions in the genome and not the genes. The genes are actually, there can be several genes in such a genetic region, and the most likely gene associated with Alzheimer's disease is then plotted here. But we for many of these genetic regions, we do not know what is actually the driver of these this association. I'm going to show you 3 genes now: ApoE4, the PCALM, and the ABCA7 gene. So the ApoE gene here is really the gene with the most effect on Alzheimer's disease. Carrying 2 alleles of ApoE4 will increase your chance of having Alzheimer's disease about 30-fold, between 15 and 30-fold. So this is really the largest impact on Alzheimer's disease that we know. Then the PCALM gene is this pile up here, and I'm showing you this because I'm going to show you that bit later in the talk. And the same thing goes for the ABCA7 here that I will also go back into further. So now there are about, well, actually very recently there's now 70 GWAS hits that are identified using GWAS. And but for a large proportion, we do not know what actually the driving variation is for those associations. When we make a polygenic risk score based on these SNPs, based on the 40 SNPs, then we can actually pull apart the 2 extremes, so the Alzheimer's disease cases and the cognitively healthy centenarians. And you see, of course, here that carrying 2 alleles of the ApoE4 SNP, or the ApoE3/4 allele, or the two ApoE3 alleles, or 2 ApoE2/3 alleles, that really is very, very directive for your polygenic risk score. So that has a really big influence. If you and then you can see that the AD cases have a higher polygenic risk score compared to the Alzheimer's, the cognitively healthy centenarians that we analyzed here. If you take away ApoE, then the remaining genetic variants still have an impact, and you can still pull or see 2 different distributions for PRSs of these individuals. However, if we really want to predict who will get Alzheimer's disease and who will not, we need to get rid of this overlap here. I'm not sure if we can actually really pull them apart completely, but I think we can pull them apart more than we can now, because we need to know which drivers of these genetic associations are, what is driving this. If we test the drivers instead of a proxy for the driver that is in linkage with it, then we probably can pull these 2 groups apart, so that we can actually use this for risk prediction. An interesting paper that was published recently was by Zhang et al. And he writes that whereas most diseases have maybe 200 GWAS SNPs, so because GWAS is done in all kinds of diseases, but usually in many cases, lots of these genetic loci are identified, maybe 200 of them, for example. But with Alzheimer's disease, we don't seem to get too far. So we now have 70, but we would have expected a lot more of these loci. But so Zhang et al. Suggests that Alzheimer's disease may be an oligogenic disease, so that the GWAS loci that we have now identified, that's where it should be. We should really fine map those loci, because something in those loci is actually causing this disease. And it's not the 200 GWAS loci, but maybe it's only 70, or maybe in the future 80. But it's not as polygenic as other diseases. So to identify rare genetic variants in the genome, the field turned to genome sequencing between 2010 and 2020, and it allows the identification of very rare variants. So here, of course, is the common variants that we investigate using GWAS, but then, an unknown pathogenic variant is obviously not measured on a chip, so we cannot identify it using GWAS. With genome sequencing, we can actually read the part of the DNA, so we will also read, of course, the common SNPs in those reads, but we can also read the very rare genetic variant that may be actually risk increasing itself. So it is possible then to identify the culprit. And we have done an analysis of about 50,000 exome sequences where we compare Alzheimer's disease cases with controls. And we are identifying novel genes, also genes that were already known, of course, but genes of which a rare genetic variant is more likely to occur in an Alzheimer's disease case than controls. So for example, in the SORL1 gene, when you have a very, very rare predicted pathogenic rare variant in the gene, it is more likely to occur in an Alzheimer's disease case compared to a control. And this goes for several other genes, and we are identifying novel genes. So this is a paper that we recently put on medRxiv, so please do look that up. However, the rare mutations that we find in this analysis, they will not map to a specific haploblock. So they will map across haploblocks and will not explain a GWAS hit. So therefore, while we are understanding a lot more about Alzheimer's disease and the genetics of Alzheimer's disease using this exome sequencing for these rare variants, we still cannot understand why these GWAS regions, for many of them at least, why they associate with Alzheimer's disease. And so this is what really it comes down to. The drivers of these GWAS regions are unclear, and we have never investigated the structural variants that are in linkage with the GWAS SNP or the GWAS haploblock. And so this is the PCOM region, and this is called the PCOM region because PCOM is one of the genes that lies in this region. However, we also see that there are structural variations that map to this region. And one of these may be expanded, or there may be something pathogenic about one of these structural variations that is in linkage with this GWAS SNP or GWAS locus, and therefore it may actually explain the GWAS locus, and that we need to investigate. So now we're going to go into the value of long-read sequencing in Alzheimer's genetics. So we know from previous findings that structural variations do occur and associate with neurodegenerative diseases. We know the C9orf72 in ALS and FTD, and of course we know Huntington's disease, but there may be other structural variations that we have not identified yet. And so we've never really investigated deletions, insertions, inversions, and all those structural variations, because simply it was too difficult to identify them using GWAS. It was not possible, and using short-read sequencing, it's also very, very difficult. And one of the most interesting structural variants that we think we should investigate is the variable nucleotide tandem repeat, the VNTR. And basically, it's a repeat, in this case, GCT-GTT, that is repeated here. And here we show 3 repeats, but it can also be expanded until very, very, very many repeats. And what we know from VNTRs is that they may actually obstruct gene transcription. So they may actually have an effect on gene transcription and thereby on disease. It may be that the GWAS region, say for example that this G is the allele associated with Alzheimer's disease, is in linkage with an expanded repeat. And that would be interesting to investigate. And that's what we want to do. In fact, in 2018, Arne de Roek et al. Published a paper where they found a VNTR that was in the ABCA7 gene, so which I've shown you previously is also associated with Alzheimer's disease in the GWAS study, so it was a GWAS locus, that an expanded repeat in an intron in the ABCA7 gene is associated with Alzheimer's disease, and that this repeat is also in linkage with the SNP that is identified with the GWAS. So therefore, an expanded repeat may explain the GWAS association with Alzheimer's disease. De Roek et al. Found that when the VNTR exceeds 5,200 nucleotides, then that occurs more often in Alzheimer's disease cases compared to controls, and it associates with about a 4.5-fold increased risk of Alzheimer's disease. And Probably there are many, many more such variants. So we wanted to get acquainted with long-read sequencing, and we decided not to look into one Alzheimer's disease case. We wanted to look into a 115-year-old woman who was not demented. And this is Jasper Lindhorst. He's a PhD student, and he is the one who did all this work. And this is actually a publication that was recently came online, and then you can actually look at the data there. So this is Mrs. van Andel Schipper. She was born in 1890 and she died in 2005. So indeed, she was 115 when she died, and she was born prematurely. She had no children. And interestingly, her mother also died at 100 years, and also she was not demented. When she was 82, she decided to donate her body to science. And then when she was 112, she decided, well, you know, I'm very old now. Maybe they don't want me anymore. So she decided to call the hospital, and that led to a visit to her where it was possible to completely test her brain, how it functioned. And they found that at 112, her brain was completely cognitively healthy. So they went back the year after, and they did the same sorts of tests, and then she was still at 114 at that time, still completely healthy. At 115, she died, being cognitively healthy, and she had donated her body to science, so it was possible to collect some of her organs and also her brain. And so I'm going to show you her brain, but first I'm going to show you the tau staining that is associated with Alzheimer's disease. So tau is a protein that accumulates with Alzheimer's disease, and it starts here in the hippocampus. So this is a brain cut like this, and it starts here in the hippocampus, and then it progresses throughout the whole cortex when the disease progresses. And usually people come to the hospital when they are between the Bragg stage 3 and 4, where they say things are not right. I'm not my old self anymore. Something is wrong, and that's usually here. Now tau also builds up in normal aging, where it doesn't have that big of an effect. So people can probably be resilient to it, but there is a limit. I mean, it never builds up to these stages. But Mrs. van Andel Schipper did not build up that much tau. She was a Bragg stage 2, and well, I collect centenarians and also their brains, and the more that I know about the centenarian brain, the more that I know that this is really, really remarkable. And also, she also did not build up any beta amyloid plaques. She also did not have any vascular brain pathology, and she also didn't have any atherosclerotic changes throughout her body, which is really, really, really remarkable. So this, in my opinion, is really a bionic woman, and we should understand how it is possible that she was able to keep her tau and her amyloid so very, very low, and well, to escape completely any type of neurodegenerative disease. So we decided to sequence her genome, and we asked, are the VNTRs in her genome perhaps shorter compared to the other VNTRs in other individuals? So we decided to do a survey of structural variations. And this is Mrs. van Andel Schipper again. She, of course, has a diploid genome, and we generated 2 pseudohaplotypes of her genome. We used her heart cells as a resource of the DNA, and we hybridized 269. Actually, we sequenced 269 SMART cells using the PacBio RS2 at the time. We had an average read length of 8.3 kb, 45x genome-wide coverage, an N50 of 7 kb, and the total reconstructed genome size was 2.8 gigabase pairs. I should note that the long and the short read data is publicly available, so go look up the paper and you can download all that data. We decided to compare her genome with the CHM1 haploid cell line, which has an N50 of almost 27 megabase pairs, and then also with the CHM1-3 haploid cell line and with the reference genome. So we wanted to know whether or not the VNTRs that are expressed in the brain are on average shorter compared to those in the other genomes. So we found, of course, that analyzing these structural variations was not easy because boundaries shift between individuals. And also, so we found that Audano published a paper in 2019 who compared 15 genomes, and he used SmartSV and to compare those genomes with the reference genome, and then he merged those calls when there was 50% overlap. And then you can see that there are multiple structural variation calls in a specific locus. Now, we compared 5 genomes and we used the REVEAL algorithm, which is a graph-based multi-genome alignment. And that allowed us to identify 1 structural variation per locus. So whereas Audano, he identified almost 100,000 structural variations, we found not even a third of that. But that's because we merged all of those loci into 1. So, and that is actually also published in the paper, so you can look up that database. We found that more than 8,000 structural variants that we identified were not in the Audano structural variation list, and we found more than 3,000 novel structural variations that were unique to the W115, and this is Mrs. van Andel-Schipper assembly. And all of the details and all of the lists of this analysis you can download from this paper. So we found several types, of course, of structural variations, but we found that of all variations, the VNTRs were really most common. So 70% of all structural variants were VNTRs. And we found that in the subtelomeric regions, we found that there was a 7-fold enrichment of VNTRs, but there was a 21-fold enrichment of VNTRs that had a GC content of more than 60% and a repeat pattern of more than 50 base pairs in the repeat. So that we found very, very interesting, because why would that be? We found further, because we were interested in neurodegenerative diseases, that specifically more VNTRs occurred in long genes, but that was based on the size of the gene. The longer the gene, the easier it is to get a VNTR in it. But long genes are predominantly expressed in the brain, we found, and long genes were predominantly associated with neuropsychological disorders. So having more VNTRs in these long genes might associate VNTRs specifically with neurodegenerative diseases, simply based on the length of the genes that are expressed in the brain. And so we did an analysis. Here you have the gene length, and here the number of VNTRs. Now, we make a distinction between non-subtelomeric and subtelomeric genes, VNTRs, and you can see that the actually genes, and you see that the longer the gene in the non-subtelomeric genome, there is a variable distribution of the number of VNTRs, but those that are in the subtelomeric region, you can see that there's an association between the length and the number of VNTRs. So apparently, genes that map to the subtelomeric genome are more likely to have more VNTRs, and those are more expressed in the brain. Now here we found that the PTPRN2 gene and the DLGAP2 gene, they have really a disproportionately large number of VNTRs here. And again, these are both predominantly expressed in brain, and they are, through all sorts of different analysis, associated with neurological phenotypes. So these two genes, they were not associated yet with VNTR types of diseases. So those are not novel in this analysis. So these GC-rich VNTRs that map to these subtelomeres, they seem to be recombination hotspots, and there is lots of genome instability here. And the meiotic recombination rates are also very, very high. So since all this stuff happens at these little ends here, this may actually drive evolution. And indeed, the brain, of course, is one of the most recent, or actually the human brain is one of the most recent developments in evolution. There's many highly variable gene families that map to these regions, such as the olfactory genes or the immunoglobulin heavy chain gene and the zinc finger proteins that are really subtelomeric genes. So it could be that the evolutionary adaptations actually occur here at the tip of the chromosomes. So our original question was, were the VNTRs expressed that were expressed in the brain, were they shorter in size, or they're not expanded in Mrs. van Andel's genome? But we had a lot of difficulty answering that question because there were too many technical differences between the genomes that we measured. So the DNA resources were different, the contiguity was different. The reference genome, of course, is a genome sequence from Buck clones. So it was very difficult to really compare the lengths of the VNTRs. We saw that the PacBio assemblies relative to the reference genome had more expansions, and Mrs. van Andel, the W115 haplotypes, had also more expansions compared to the CHM1 and the CHM3 genomes. We then looked up whether or not cloning could have an effect on this, and we found that cloning may lead to shorted alleles because muted gaps or unstable genomic sequence may actually be shortened. So therefore, we could not answer this question, and we realized that we need to do a very large systematic comparison between Alzheimer's disease cases and controls to answer this question. So what we now want to do is since I work at the Alzheimer's Center in Amsterdam, we want to take a large group of Alzheimer's disease cases, and we want them to compare them with a large group of centenarians that we are collecting in the 100+ study in the Netherlands. So we have now more than 400 cognitively healthy centenarians that we want to investigate and want to compare to a large group of Alzheimer's disease cases. And one of the things that we want to do also is to generate a compendium of non-pathogenic VNTR length distribution based on this group of cognitively healthy centenarians. So to conclude, VNTRs, or structural variations, but specifically VNTRs, may explain the missing heritability that we find in many diseases, but also including Alzheimer's disease. We do not know all the genetic factors that lead to Alzheimer's disease, and the impact of structural variants was never investigated. We find that VNTRs occur more often in long genes, and long genes are expressed in the brain, so there may be a connection there. We need to do a systematic comparison of Alzheimer's disease cases and cognitively healthy controls. And the challenging part there will be that we would like to, of course, to include structural variations in risk prediction algorithms, such as the polygenic risk score. That's not going to be easy because we cannot collapse an allele into one. But so these are interesting challenges that we need to work on. We should also say that structural variants may be actionable. There are, for example, for the C9orf72 repeat, there is the Alector is designing novel drugs that may actually be a good treatment for individuals with a expansion. We see that from all the Alzheimer's disease GWAS loci that several map to the subtelomeric ends, and those might be of the best interest to investigate first. So this is the group. So we also collect the centenarians, and we also are involved in Alzheimer's disease genetics. So we really want to compare both, and these are the individuals who are involved in all of that. We are hiring at the moment, so we have a PhD position for bioinformatics on this comparison of Alzheimer's disease cases and centenarians with long-read sequencing, and we also have an MD PhD position open for the impact of rare variants in families, and we also have a group position open. For a supportive function. So please come look that up. I posted it on my LinkedIn and on my Twitter account, and you can also email me at this email address. With that, I'd like to show the people that I collaborate with and my acknowledgement slide. And these are the individual. Oh, yeah. This is where I'd like to end.